So just bear with us. Right now in the screen we have some sponges, some of those yellow corals again. Is that another slit shell in between, basically,
No,
in between the little crevice there?
It's a fish. Sorry, it's a fish.
It's a fish. Oh, okay. Roland sits, our video engineer, sits over in the corner with the really nice high definition monitor, so he can often see things way quicker than we can.
Yeah, Roland, I'm wondering if you could tell me whether you think that the material to the right of the boulder that's in the center of the screen looks like there might be some loose rocks there on your beautiful high-def screen.
Actually, I don't think it's loose, but that one that you're looking at, it's got sort of a stick coming sort of between 3 and 4 o'clock. That one would be the most likely culprit. If we get the pilot to rotate over there, once they do, we can zoom into it, but that one would be the most likely candidate. That or the one in the bottom right-hand corner that looks darkest with a piece of white coral rubble laying on top of it.
Right.
A quick shift change.
Yeah, I see what you
Right
mean.
with you.
So guys, just interestingly, in the Sirius view, you can see the sort of the edge of the platform or whatever it is we're sitting on. So you can see all of the gorgonocephalid basket stars sort of lined up along the edge, putting themselves into the currents, like I was suggesting earlier.
The interesting thing about Sirius's performance
Yes,
today
that's
is
true.
as the currents change, she's done like 3 of these.
Thank you, Diva. And you can't quite see it now in the Sirius view, but a little bit earlier there is a shelf
So
just below
really
that, and there's another line of them all lined up right along the edge of the shelf.
Okay.
Interesting. When I sat down, I was right over the top of D2. I'm moving behind it.
Pilot, this is
Pilot,
watch
I
one.
think the current's roughly from the south because the tether has been blowing that way. Certainly not from the north.
Stand by just a second, watching.
No problem.
Okay. All
So,
good.
Yeah, we're settled out
Okay.
as much as we can be. Current.
Hi, watch lead pilot.
Hi there, Pilot. Yeah, we were wondering if it might be possible to see if on the right side of the field of view on the seafloor there's a dark-looking rock that might be loose. And we haven't picked up a rock here. We've been looking for someplace, and most of them look like they're totally attached, but that looks a little bit loose.
Down here at like 5 o'clock, coming up to like 3 o'clock right now.
Exactly.
Okay.
That's right.
Look,
There's one right in front of you. Okay.
Maybe
Yeah, if that is pick-upable, it would be great to pick it up.
pick upable.
Yep, that's a technical term.
Oh, okay. I hadn't studied that. Okay. Oh, well, we seem to be in a good position here, so
Yeah, thank you.
let's see if we can get this rock.
All right.
Patty, this is Deb. Where do you see a loose rock? What am I missing?
Okay, Deb, I wonder if you're maybe a little bit behind, maybe 10 seconds off or so. But there is, in the center of the screen that I'm looking at right now, there's a cluster of dead polyp bases, and there is a small-ish black rock. Yeah, the black rock. How does that
Hey, now,
look to you?
Co-pilot, do we have any rocks in
Oh, all rocks look
the left wing?
Haven't
Tonight,
turned on the
certain.
craft.
Oh, okay. Which box would you like to stow it in?
Well, might as well put it in the left one.
All right.
So a good steady place. So you'll bring the wing in, I'll flip
Yes.
it open and that
Do we
kind
know
of
how
thing?
Yes.
big it is?
Okay.
We have,
There's lasers.
yeah, we have the lasers on. It looks like it's about
Yep, we are up. and
3
running.
inches. It is kind of small, but we've seen so few rocks that look as if they might be loose and accessible.
Great, thanks. Okay, hydraulics coming on.
I think I'd rather not wait and see if we can find something else because we haven't seen very many.
I totally agree. I just hope that at some point in time we might try like one of the little overhangs.
I agree. Yep. I have not forgotten you or it.
Looking up something here. Yes.
Deb, just for your information, in case you haven't heard, the pilots, there's a real difficulty with going off the top of the ridge and down just a little bit to get under some of these ledges because there's a ripping current that's going through. And what they are concerned about is they get close enough to the vertical wall, to pick up the sample. They can't open the box. They can't swing the sample box in to get it in position. So what they'd have to do is grab the rock, hold on, and then if they back off the ridge, they're going to be blown up into the water column. They're afraid they might lose the sample.
I was thinking less of those big ledges and more when we saw those sort of angular holes in the rock. Maybe we could grip one of those.
Okay, we can try that.
No idea if we really can.
This rock looks like it's pretty well welded into this substrate.
Yeah, that does not look like a rock that's not in place.
Yeah, pilot, I'm sorry. This one looks like it's going to be a no-go. It's pretty stuck there.
We had another one over here that looked interesting.
Ooh.
Tilt your camera up.
Sure, thanks. No. These are all welded in.
They do look like they're really glued down.
Yeah.
And it.
Yeah, those aren't moving.
Deb, that makes me think that very likely this is ash that was warm enough, when it fell, to weld all of these additional rock fragments right into the matrix of the ash fall.
That makes a lot of sense.
Okay.
Yeah, despite the fact that
We need to adjust the shilling.
well, maybe we'll find a ledge that has really thin shoulders sticking out that could be knocked off by the manip.
Okay.
And we could try that later too.
I hope so.
Yep, me too.
Yeah, that'd be great. Thanks anyways.
Oh, you bet. The pilot, so what Deb Glickson and I have been thinking about is sometimes these little local promontories, little ledges that we see have something sticking off of it that might be able to be broken off with a manipulator if it's not too difficult to do so. And even if, we get a small fragment, that still is going to be something.
Okay, understood. If we see another, although these look very likely, and obviously they're quite glued in, but we'll keep our eyes open.
Okay, thank you.
Great. Okay, and we've had a shift change up here in the front row, so we have a whole new set of people from Global Foundation. So, Levi, why don't you do the intros for us?
That would be great.
Hello there to all our listeners. So yes, the front row, we just changed out for the shift change halfway through the dive. I am the navigator for the group here, Levi Unima. We have pilot Jeff Williams and co-pilot Dan Rogers and video Tara Smithy.
And our video clipper is Anna. Anna, why don't you introduce yourself?
Hi, I'm Annie, and I work with the GFOE.
Thank you, ma'am.
You're all secure.
So, watch lead pilot.
Go ahead, Pat.
What? Where are we going? What do we do?
Yeah.
We want to head toward our...
Wait, don't go anywhere yet.
Don't go
Don't
now.
go anywhere. I want to just ask, where are we? So I just wanted to ask the rest of the scientific team what they want to do about this one collection here.
Broadcast that on OKEX.
So science team back on shore, I just sent you a message about collecting the Spongoceroides. I've been spending some time looking up information. There is Spongoceroides has never been reported from the Pacific, so it's definitely a new record of occurrence for both the genus and the species. So I don't know if we want to collect this or not. Yay, nay?
Now, which one is that?
The one
Shirley?
that
The
lump
with
of
the
yellow stuff that has the vermeted gastropods in it. Okay.
Shirley, was that the horrible thing that looked like it had worms?
Yes, that's the one.
Okay, thanks.
Where was that?
She said, looks like it has worms in it. Okay. Okay, watch lead pilot.
Yes, go ahead.
Okay, so you see those kind of lumpy yellow things that had the worm tubes sticking out of them? It's
Kind of like right dead center right now.
On the left?
No, to the...
On the left side of the screen?
Yeah, no, to the left. At 9 o'clock, all the way over.
So it looks like a clump of Jell-O sitting on the seafloor, sort of?
Yes, probably more. I would
I'm
say
sorry.
Jell-O as a kind term. I'm sure that everybody else is thinking something more disgusting than that.
You mean like a cow pie?
Yeah, a yellow cow pie.
Okay, I see what we're talking about.
Yep. There
Oh,
you
there's
go.
one on the rock behind too.
And it's common.
Very...
It's common at the. And it usually occurs at this depth in the Atlantic as well. But as I said, this genus has never been reported, as far as I know, never been reported from the Pacific at all. And
Wow.
This looks remarkably similar to the species that we find in the Atlantic and Caribbean.
So this is a sponge-like.
It's sponge,
critter?
yeah.
Okay.
It's going to be. It's going to be kind of, I don't want to say brittle like glass, but it'll crumble, and we don't need the entire sample.
So you mean you don't need the entire organism, just a piece of it? So if the fingers of the manipulator went in like on the right-hand side and
Right.
got some of the tissue is what you want?
Yep.
Okay.
All right. Let me just settle in here and see. But Dan, I think we'll do the same thing as we did yesterday, where we take the sample and then come up and open the bio box.
Okay. We can do that. Which box would you like to put it in?
Oh, the easy one.
All right.
Port side.
So we're at a ROV depth is 359 meters. This sponge looks to be about
Am I okay for hydraulics?
Hydraulics are up.
17 centimeters
Yeah,
in
about
diameter.
That. About that.
And it looks like it's sitting on
Take your cameras.
a surface that's primarily
Okay, thank you.
a volcaniclastic accumulation on the side of a subsidiary vent or cone on the side of Maug Island.
This sponge will be of a lot of
Group
interest
force 4.
once we take a subsample of it for molecular work, because we'll be able to compare it with other sponges, other Spongoceroides siliquaria from
other
the
parts of the
Atlantic.
world?
Yeah.
So the pilot's going to touch it first to see sort of what the consistency is like. Looks pretty hard.
So the little tube things, the little
On the
shells
top.
on the top, all over it. Actually, it'll be throughout the sponge. Are vermeted gastropods, and the species name is, at least for the one in the Atlantic, is Siliquaria. So Spongoceroides siliquaria, and the siliquaria is the name of the vermeted gastropod that grows inside the sponge.
Now, wait a minute. This thing has a gastropod inside of it?
Uh-huh. Yep.
So what is it doing?
It's like real estate.
Real estate.
It's an unusual commensalism, honestly,
Really?
to tell you.
So,
yeah.
So they always occur together.
Well, there are other Spongoceroides that
wow,
that
this is hard to
don't have
sample.
vermeted gastropods. Yeah, it's kind of crumbly.
We need a scoop. Oh, do you think it's coherent enough to pick up those pieces?
Yeah,
Just the pieces that got
I can try.
removed?
We can try. It is crumbly.
Yeah, if it's possible to pick up the little pieces, it's amazing. The pilots here have this incredibly delicate touch with the manipulator. They know how much pressure they're putting on the fingers of that arm. So it's up in the water column now. I'm going to get a look at it as the pilot rotates the manipulator, and
or if you just would prefer to get it in the box, we can
I
do that.
think we better just get it in the box.
All right.
Yeah, then everybody hold your
All right.
breath.
We can get. I think we can get the drawer.
Okay, give it a shot, please. That's very delicately held there. Oh yeah.
The bio boxes are just being slid out,
There
and
you go.
the pilot's going to try to very carefully move it over to one of them and plop it in. And a round of applause.
Yeah, very nice.
Round of applause for our pilot. Great job.
Port and thank you very much. Port and board.
Thank you so much, pilot. That's great.
Sure. You don't happen to see a rock here, do you?
Oh, I don't know. There is a little rock right next to the sponge. You think that's
Tilting down.
loose? If that's a rock. Is that a rock?
That might be.
Looks like it might be a critter.
Oh.
Whatever it is, it's loose.
Oh,
fish!
That's a fish.
Oh, wow.
I could try for that.
Boy, that fish came by just when we're doing something else.
All right, we're getting a rock sample here too.
Is it a rock?
Piece of the rock.
Hold in there.
Frankly, if it is a rock, it doesn't look like.
That was 359 meters.
Okay to zoom, Pat?
Yeah, please zoom. Watch lead, is this interesting, or should I just put it back where I found it?
I find it
look at that fish.
of interest.
Okay.
Thank you so much.
All right.
And
Dan, if you bring my starboard
And
wing.
we can zoom on the
Right.
fish, perhaps, video
awfully
while
small
we're...
one.
It is an awfully small rock.
It
I
is.
think it might be worth putting in the.
Oh my
port wing.
God! Look at that.
I
No.
don't want to try to open
Okay.
a port
All right.
wing. Yeah,
All right.
that's the problem. I can't
Look
really
at that
open it.
beautiful, beautiful imagery. Wow, 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 wow.
Watch out, buddy.
Oh, Oh, don't squeeze the fish.
Whereas on this end, I'm looking at it saying, fish, don't get in the way of the rock sample.
Yeah, that's exactly what I'm thinking too. But we do want to get some nice pictures of that fish.
Okay.
So rock and starboard.
We're Bruce and I are talking about this right now. We're trying to figure this fish out, actually.
Okay, great.
Showing the wing.
Okay.
Thanks. Well, that rock looked like a piece of the volcaniclastic material that makes up most of the slope.
All right.
So that is representative of the dominant rock type. Now, where's the fish? We need to pop into the Sirius view to see where he went.
That looks good there, Dan.
Yeah, looks okay. Let me rotate the wrist a bit. There you go.
Yeah.
That looks good.
All right.
Securing hydraulics.
Hydraulics off here.
Not yet. They're
That was a
looking it up.
twofer.
Yeah, nice. Camera's back to you.
Oh, the fish is not in the Sirius view. Thank you for that twofer, pilot.
You're welcome. And I suppose we can be on our way now, unless the fish comes back.
Indeed, unless the fish comes back. But maybe he went out in front of us.
That'd be nice. He could just lead us.
Co-pilot, was that biological in the inner or outer box?
It was in the inner, the port inner.
Thanks.
Actually seems as if...
Okay, we're working on this fish. We're going to give you an update as soon as we've got it figured out. We have a lead on a different species here.
Yeah.
Thank you.
It's like...
So eventually we'll get a name for the fish, folks.
Just checking my sea bias here. Okay. I guess we push up the hill.
All right.
Okay,
let's see. So
we think this is not
see what point two there.
Edalus coruscans and onaga because of the tail on it. And we just, Bruce remembered a species of Edalus called radiosus, and we just Googled that fish, and it looks like it's a dead ringer for it. Its distribution is here in the West Pacific, and I've never seen it before, and I don't know, Bruce, if you've ever, Bruce has never seen that species before either.
Oh, man. What a dive.
I don't know if there's any in situ imagery of that species that exists, but that was pretty exciting. It had us stumped there for a while, but we think we've got it. So great shot, guys.
Estellus radiosus. It sounds like a charm from Harry Potter.
Yeah,
All right,
Estellus
so
radiosus.
HIPAC says our bearing toward waypoint 2, roughly where we're pointed, so sonar.
This is an incredibly
So
diverse area.
Oh, yeah.
Really
Point a
diverse.
little
It's
bit more
so
starboard.
different from what we saw yesterday.
Now, did we pick up one of these yellow corals that folks
no,
wanted?
they were having... I
or would they
think there was
prefer
a
the
problem.
sponge? Okay.
I think there was a problem in collecting the... You weren't able to get down on the base to collect some of the coral.
Okay.
Yes. Oh, understood. Okay.
From my understanding, there is no interference with any of the ROV sonars or anything by using the EX16s, but if there is, please let someone know and we'll try to work.
Okay, understood.
We've worked with them in the past, just for your awareness.
Yeah, thanks.
Boy, there are a lot of these yellow corals right here.
Yeah, I know. So we weren't able. You're not able to collect those corals at all.
Yep.
All right, let's go ahead and do 30 meters.
Okay.
140.
Okay, I'll call it in.
We did not grab the corals.
Too hard to grab?
It was, yeah, so when we were first setting down to collect them, the drawer, the way that the topography was, the drawer couldn't come out. And we were in such high current that we couldn't pull.
Okay.
Oh, we have another fish coming up.
We do. We do have another fish. We'll get a close-up of that.
Partial zoom on this?
Of course.
Sort of looks like a moorish but it's the wrong color.
What color?
Yeah, it's like a
black
dull,
and white.
It's like a dull Moorish idol.
They also have really long defined.
So we're getting good imagery of the rocks. There's a small spherical sponge. We can see.
Oh yeah, good image of the rocks.
Coral and live coral.
Actually, these rocks look exactly like the rocks around Hanauma Bay in Hawaii.
I've actually been there.
You have
I've
indeed.
snorkeled there.
Yeah, good for you, because it's a gorgeous place to go. But those rocks are formed by what's called a tuff cone. Man, I'm really losing my voice, lady.
Oh, no, don't.
And
I wonder if this is a spot to try to grab one of these scleractinian corals. Looks like there's an awful lot of them around that are in clumps.
yeah, and they're loose, right, Chris?
You want to try and do that, Chris? That's what you're saying?
Yeah, if you see a nice big clump, I don't know if we're further enough up and that the pilots feel comfortable opening the box. But I'm just noticing there's quite a lot of them in this area, and there are some pretty large clumps a little further up maybe, just to keep our eye open for,
okay.
for that one.
It sounds like we're about to do another sample. It sounds like, yeah.
Yeah, watch lead pilot.
Yep, go ahead.
Yeah, it looks like our shore-based science team would like you to try again to get one of these yellow corals or a clump of yellow corals if you're able to get to a place where you can extend the box.
Okay. So, Dan, maybe you stick with me for the next 10 minutes?
Sure.
I think what we'll do, again, we'll do the sample, and then if I have to lift up, I'll lift up and you'll open the box.
Sure. Hey, Nav, why don't we go ahead and bring this ship to an easy stop?
Aye, aye.
Thanks.
Bridge, ROV nav. Can we have the ship come to a nice easy stop right here? Thank you, Bridge.
That's a nice clump.
Our shore-based scientist pilot says, that's a nice clump.
Yes, but is it pick-upable?
That's the question.
I know. Actually, yeah, my part, let me see if I get not quite.
So I think these are basically loose. I think if you just sort of almost forked it as though you're scooping up from underneath with the claw, it might come right up and might actually be easier to grab intact than you might otherwise think.
Pilot, were you able to hear that transmission from Chris?
I did not pick that up. Let me just...
Okay, what he's saying is the clumps themselves look loose, and that if you orient the fingers perpendicular to the clumps, the probably be able just to scoop it up.
Okay.
Well, you can try that.
That's right. That's right. That noise you just heard was not problems with transmission. It's simply a problem with my vocal cords.
Oh, no, you better go get something to drink. Get a cup of tea or something.
All right. Tara, let's zoom in on this clump. I just want to look at the bottom of it a little bit, see what it looks like.
Really nice.
Good, hold that. So you think if I grab this thing, well, I'll give it a nudge first and see what it does.
Yeah, it may not be firmly attached to the bottom. You may be able to get right to the base, but yeah.
Okay.
You don't need an armchair collector.
Okay. Let's see what happens here. Okay, Dan.
Okay, you've got hydraulics. I'll take your cameras, switch your monitors.
Okay, thank you.
Do you want to go for the port outer?
Yeah.
Okay.
Sure.
All right.
Tara, I might need you to pull out a little bit on the main HD. Thank you. Okay, so that's what I'll pick up, that piece right there. Does that sound good to you, Watch Lead?
Yeah, as long as there's something live on the other end of it.
Okay. I'll try to make it so.
Oh yeah, that's looking good. Looking good. Did you get it? No. Good.
Pull wide, please, Tara.
Assume you just want to get it in the box?
Yep. Let's
All
get it
right.
in the box. Dan, if you could tilt me up a little bit, please.
Sure. Oh, George just nudged you a bit.
I saw that.
Yep.
Okay.
You all right?
Yep, so
Okay.
far. All good.
All right, tilting up.
That looks really nice. It looks like there are at least 1, 2, 3, 4, 3 or 4 coral, live coral pieces on there. Okay, so that's going into port outboard. Nice. Very nice. Thank you.
Well done, pilot.
Okay.
Well done. Thank you so much. And again, I think there'll be some people very grateful for that specimen.
All right, I think we'll be stable.
So is that a dendrophilia?
So we just had this discussion with Andrea Quattrini. I was calling it Aguchisomnia, and she was calling it dendrophilia. And then she went and Googled what she was thinking about, and it turns out that Aguchisomnia was originally in the dendrophilia, and then they broke it out as a separate genus. And so now it's the Aguchisomnia, and I think she agrees that it's more likely that than a dendrophilia, but it's in the family Dendrophiliidae.
Okay, thank you. I must admit, I've never heard that genus name. I'm looking up in the log. Tell you what, I'll have you, one of you on shore, actually type that into the chat room log, please. Aguchisomnia. Aguchisomnia.
Thanks, Dan.
Yep.
Smooth.
Okay.
Well, remember to keep an eye on serious thrusters today in case we
Yes.
get off.
Yeah.
Trying to understand that source of that
I
gas.
think your hypothesis from this morning is probably pretty accurate, but
Yeah, yesterday was a pretty heavy day for serials. Okay, now we'll go up the hill.
all right.
Roger that.
All right.
Okay, folks, we're
Hi,
heading
Faye.
on up the
River.
hill.
Yeah. So uphill to you. Let's see.
Well, let's see. I think if I pull up here a little bit, we can see.
Yeah.
Oh, wow.
Probably
Look at
to
all
the
these
left.
critters.
Oh my
They're
God.
everywhere.
Yeah, this was a good collection. Good collection idea.
So,
Dominant
yeah,
fauna.
I'd say
Wow,
for
wow,
me
wow, wow.
right now,
It's a
it's,
forest.
170.
170.
Yeah.
That's
And a Guccisamia forest.
A Guccisamia forest,
roughly the right
indeed.
direction, according to HiPAP. So
Guccisamia.
yeah.
Never
Let's
heard. Okay, we're also seeing
if
some
you wanna come saddle
astrophorid
starboard, we can
sponges.
Okay.
call in a 170.
You got it.
Is the white thing on the
On
right?
the on the coral.
it's 30 meters.
Yeah,
is
looks like
that
it might be
a-
a,
30 meters.
Looks like it might be a sponge also.
170.
Tara, let's take
Different
a
sort.
Go ahead and call it in.
partial zoom
Okay.
on the
Indeed.
The sponge?
white thing coming into.
Roger.
Bridge RV nav. We are ready for a ship move now. 30 meters, bearing 170 degrees, decimal 29.
Oh, Tina said it's a meadow. It's a
A
meadow because they're
I
like a meadow
love the
of,
word.
of daisies or. Oh,
Good copy,
I like
Bridge.
that.
Very sweet.
I like that. And the Guccisamia meadow. Typing that into my notes.
There's a nice image of one of your sponges.
It is.
Okay.
Could be a
We'll wipe this
geodid or it could be a pacastrellid, but
Cross here and get in front of
Not
Dan's
sure
view
what.
a little better.
I love that name, geodid.
geodid.
Like a geode.
That's
has nothing to do with a real geode.
No. But if you cut it open, the reason it's like a geode is
It's 'cause
because
there's
it's got that-
stuff inside
No,
that
there's a
looks like-
thick cortex that's loaded with spicules, and then
Yep.
it goes.
Now, are they radiating in toward the center of the sponge?
That particular one does not, but there are some
Are they
that do.
just
There are
randomly oriented?
No, they're oriented in a certain pattern, but it's not
Okay.
radiating out. But there are some spherical sponges for which the spicules do radiate.
They do radiate? Boy, that'd be pretty to
And
see.
so how the spicules are arranged in the sponge actually helps you to diagnose what it is.
Oh, no doubt.
Okay. That was a comatulid crinoid that we just passed over and got some nice imagery of.
Man, look at this.
I know, amazing.
It's just so uniform. Wow. And there's a lot of stuff floating by in the water, too.
Yeah, the midwater transect has
It's going
potential
to be looking
very
good.
interesting.
Oh, yeah.
Has the potential to be pretty interesting.
Shall we give a prize to the person out here who counts the most number of things
in the water column?
In the water column?
I'm not going to enter that contest.
No. I know where there's some
It's
candy.
like, it would be like that thing. I would have a code for that. TMTC.
TMC.
Too many to count.
Ha ha ha.
What were you saying?
And what is that sort of brown blob at the top of the screen?
I don't know.
Just went off the top?
Yeah, I don't know.
Oh, cute.
Look at that
It's
little
a
hermit.
hermit crab.
Little hermit crab.
Boy, our videographer has such an eye. And just look at the incrustation by biota of the surface of this seafloor. I can't.
Oh, and that grayish stuff is an
Yeah, can
encrusting
you zoom on
sponge.
its,
all the pale gray,
And
soft,
pale gray, yeah.
and pink. The pink stuff next to it at the bottom of the screen.
that's
That I don't know.
Could that
It could
possibly be an alga?
No.
Pretty straight.
Well,
I don't think it is. I
No,
think, if
guess not.
anything, it might be a tunicate.
Octocoral we were seeing.
Well, was it? You think it is that? By the way, that Stoloniferous octocoral was a royal pain to scrape off of the rock. I did my darndest to get some. We've got good material for the molecular work, but I seriously doubt if anybody would do any kind of structural work on the samples that we got off. I mean, they were like a millimeter or less. I was blessing all of you yesterday for those darn Stoloniferous octocorals because they were really tough to get off of the rock. What is that?
That's what I wondered.
Surely that makes it even more valuable because you can imagine how few there are available.
Well, thank you.
It's hard to get.
That, I don't know. If we get,
like
can we get a closer view? It looks like a rock.
It's like a rock. So I'm going to channel a geologist right now. Anyway, Deb said that she had a nice email exchange with Gene Ranke at University of Kansas, who's our carbonate geologist, and he thinks that the rocks at the beginning of the dive might have been carbonate. They look biological, so perhaps covered with. Well, he says perhaps covered with algae, but I'm not so sure about that. He did agree that the bedding we saw during the crab fight did not look like carbonate.
No way.
And they were both wondering. They were both wondering what the rock was that we just collected.
Well, I have a feeling that that's just a piece of scoria that was associated with this. If you look at the detail,
Is it scoria that we're looking at right now?
Oh man, you know. I can see little chunks of other kinds of things in it.
30 more eye.
So it could be, but then just to the left of the rock, there's something that kind of looks
we'd
like,
like to extend that last move by
it's
an
the
additional 30
base of one of these yellow
and that was bearing
corals.
170.
Because I was thinking, maybe this is all coral, but no.
Thank you, Rich.
It's just the material that is left over from a coral having broken off.
It's going to start flattening out, right?
Yep.
So this might just... Yeah, why don't you push out ahead here, in my view?
Pushing out ahead.
Might get us just on top of this ridge. This little ridge here. See what direction that goes.
This is some more of that odd-looking, odd-colored rock.
Yeah.
Okay, so we're seeing some crinoids here, the comatulate crinoids.
Those are the red-brown things. And a lot more of these little solitary yellow corals.
And the name of those?
I forget.
Aguchasamia.
Aguchasamia. Well, maybe if I write it
Think
down,
of like
I'll remember.
Aguchigami. Think of Aguchigami. Aguchasamia.
To follow.
Yep. Let's
well, there seems to be more live ones
readjust
here.
that ship move nav. So bring it to an easy stop, and then we'll get it dialed in.
More live Gucci's.
Looks like there's, even though it's pretty flat, there's still a bit of a ridge here. So.
Bridge ROV now. Can we bring the ship to a stop? I think we might change
this is nice.
This is amazing. Nice, she
My impression
says.
here is that we're getting to the top of a rise or a local rise, and the corals are more abundant on, or the living coral is more abundant on the top than the wall we're just climbing. Is that backed up by the bathymetry that you're looking at?
Absolutely, you're exactly right.
On the nav screen.
Great.
Yep, we are just now getting up to where this ridge is starting to flatten out after we were climbing the steep base of it. So most of the rest of the dive is going to be on the top of this ridge.
I was going to note this earlier, but I wasn't on the phone. When we were looking at all of those basket stars that were on the edge of the ledge of the wall,
we have a ship
this is another good example of it, and that is the effect of the change in topography on the currents. And a way to think about it is to compare it to the wing of an airplane, which is curved, which accelerates the air flowing over it. And for all these organisms that are suspension feeders, meaning they're getting their food out of the water that's floating by, they want to be where the water is moving quickly because there's more food per unit time. So it's not too much of a surprise to see more of these suspension feeders alive near the top or the maximum curve or angle of this topography.
Thank you. Absolutely. You're
and Tina Molitsova from Russia, from the Shirshov Institute in Moscow, has commented, because I think I may have even said this earlier, that these might be solitary corals, but they're not. They're pseudo-colonial. Okay, so they form a colony, but the individual polyps are not connected. So that's why we see several of them kind of in that same clump. Just like Lophelia.
So I was going to ask Chris about the precious coral distribution on a ridge like this, is there a depth constraint on them, or should we expect to see some of them up here as well? Yep.
Yeah, so the precious corals generally extend all the way up to less than 100 meters. There's precious black corals that are in the mesophotic zone that actually can be found at 40 meters depth. But this particular range would be considered more of the deep precious coral range, and that's generally about 350 meters to 600 meters.
Thank you, Chris. Okay.
Getting to the end of your tether.
Okay. Tara, let's zoom geology.
Forward lounge.
What about that rock right there? Do you think that's loose with the lasers on it? That looks pretty good.
Okay. Go ahead. I think that I've got the forward lounge. Can you hear me?
Holding pressure.
Do you like that rock?
Oh, sorry. Yes. Whoa,
You
big
like that
rock.
Okay. Do you like it? Oh, what's all that over on the right-hand side?
Yes, we want the rock.
Want the rock, okay. Copy, they want the rock. Let's
Yeah,
bring
Well,
we
this
that's
want
all
the
that
rock.
stuff. And then after you get the rock, I want to see what's on that other
Are
thing.
we, how much is left in that move? Because we might be able to just finish it out and give you some room to push off when you... Hey, Nav, I can't hear you.
He was muted. Bridge. Okay.
Yeah, we could actually put that in the starboard rock box too.
Rich, can you
'cause
bring the ship to an easy stop, please?
it'll be quite different from the other one.
And bigger.
Yes, sir.
This is a beautiful area that we're seeing right now with a meadow of daisies or dandelions, and those are corals. They're pseudo-colonial corals. Their scientific name is very strange, Aguchasamia. And as we've gotten up to the top of this ridge, we're seeing a lot more live colonies than we saw a little bit deeper. So we're getting towards a kind of an optimal depth range for these corals. This has been a really interesting dive today, seeing a lot more biological diversity, certainly in the macrofauna, the larger things, than we saw yesterday. Yesterday was quite interesting as well because there were a lot of different bottom types, and with each transition from one to another, we saw different biological communities. And today is really, really interesting in terms of the fauna that we're observing. So we haven't seen any of the fan octocorals, any of the bamboo coral as we've gotten a little bit shallower. We're at 346 meters right now. So the pilot's got a really tough job because he's got to keep the Deep Discover ROV in position. And at the same time, he's opening up the one of the rock boxes, and now he's going to deftly handle that manipulator and go down and pick up our second rock sample.
All right, Dan, tilt up a little bit so we can get a
Tilting up.
good light on this.
Holding.
Okay. Tara, do you want to zoom in on that a little bit?
Oh, wow, that's nice. Got some hydroids on it as well. Looks like polychaete tubes. And no, I am not going to be trying to scrape those little boogers off.
It looks great. It looks like a fantastic rock sample.
Good. Okay, video, as soon as you're happy with that, we can go ahead and deposit that into the rock box.
Alrighty.
Thank you.
Okay.
Okay. Just make sure the lid closes here.
Yep, go ahead.
Perfect.
Beautiful.
Looks good.
Okay, great. Terrific.
Craft is secure.
Thank you. So can watch lead pilot.
Go ahead.
Yeah, so there was a clump of something just to the right of the rock you picked up. I'd like to just get a view of that, please.
Clump to the right. You got it.
Yeah. Yeah, just below and to the right of the right laser dot. Below the laser dots. Yeah, sorry. Yeah.
Oh, I was already there.
Yeah.
Okay.
Just get a
That fell
quick
right
zoom in there. Yeah.
Okay. Go ahead, Tara.
Ready, pilot?
Yes, ready.
Okay. Any ideas on this? Are these, boy, I have no idea. Are they zoanthids? Shore-based team, any idea what these are? Wow, look at that. Oh.
I would have guessed sponge, but you're saying
Yeah, you know, now that we're close to it, yeah, it is. It looks like it's a sponge. Sure does. Now that we're close enough to it, yep, that's a sponge.
It's overgrowing one of these colonies of the
boy.
sclerotinia.
Yeah. Yep, cool. No idea what that is. Demo sponge, but not sure what. I wouldn't even venture to put it into an order. Beautiful. So in this we're seeing the coral, a sponge,
also,
and urchin.
Bryozoan overgrowth on that one piece of the coral.
Yeah.
That's sticking out through
Yep, I see that bryozoan on it as well. Very cool. Okay. Thank you, pilot.
Clear.
Give you an idea how big it is? Okay. Right. So, hi, Don.
Hey.
How are you?
I'm doing fine. How are you doing today?
Good.
Good.
You're looking forward at me, so I think we'll get the ship underway and then I'll get moving.
Okay, roger that.
So, Richard, if you would like to call in the move again, hopefully this time we won't change our minds.
Hey, watch it.
Hey.
Were you looking for
I
another-
heard that.
Same as last time, 30 meters at 115.
So guys, how much time do we have left in the dive? Do we have about 30 minutes left? And if that's the case, I wonder if
No, we have an hour and 30
cover
minutes.
a little more ground so we survey the top of this ridge a little bit more, since I think we just got to the crest. Is that right?
Yeah, we have probably about an hour and 20 minutes.
Oh, we have that much. Okay.
Okay.
I didn't realize that.
Hey, we're booking.
Bridge, ROV nav. Bridge, can we continue on the course?
It's pretty interesting to see if this Aguchi sounding extent covers the entire ridge top, or again, if this is just a patch on part of the ridge.
Yeah, we ought to be able to see that and the way we're heading.
Good copy, Bridge. Thank you.
Yeah, watch lead. I meant our collective minds. I did not mean to cast any aspersions over the back wall there.
I can't talk. I'm laughing so much. Okay, 345 meters. Very weird sponge. What the heck was that sponge? Very weird sponge. Thin encrusting. Okay, we are at 345 meters, proceeding along our transect towards the top of this ridge. Sorry, Tina, I'm not sure if we're talking about this. You're asking me to zoom in on, yeah, I'm not sure which coral you're asking me to zoom in on. I know you have a little bit of a delay.
I wonder if she's referring to the large white one in the right background.
Oh, like in the distance, like kind of a fan thing all the way in the distance?
Yeah, I'm just guessing.
Yeah. Okay. Yeah. Yeah, pilot, just continue on this course because it looks like there was some kind of branching organism that they wanted to take a closer look at.
Roger that. Okay, Tara, let's take partial zoom here.
Okay, so it's looking like it may have been a corallium, dead now. Okay. Okay, video, as soon as you're done with that, we're fine.
That was what Tina was suggesting, but I'm not sure this is a corallium.
I'm not so sure either. I think it looks more like Enelopsamia or another dendrophiliid.
Yeah, I agree with you, Chris. I think Tina was just making a guess from a distance. It was a good guess.
Yeah, and I'm thinking that it probably wasn't the one she was talking about because she's got a little bit of a delay, so it may have been something that we saw sooner.
Maybe another one of those sponges we had a look at a few moments ago that was going around the skeleton right in the middle there.
Yeah, could be.
There's a fish under that, just off to the right of center.
We see a fish?
Yeah, Tara.
Is it like the little one underneath that coral?
There you go.
Oh, yeah, there you go.
Hold that.
Hello,
cutie. Nice.
Beauty. Oh, he's scared.